The title and what I'm going to talk about is specifically in terms of thinking about increasing yield and, in Miscanthus, but sort of stepping back really for a moment. There's thinking back to some of these big issues that we're facing at the moment, and particularly the massive challenge in terms of how we're going to respond to the elevated concentrations of CO2, particularly in terms of what the believed consequences of those of that increase will be. And if we look at that in terms of the challenge that we have, in terms of the trajectory on which we're on, so that's this dotted line here, and really where we need to be in terms of actually stabilizing CO2 emissions. This size of this task is basically too big from the perspective of any one sort of renewable energy or carbon-saving technology. So whether we're thinking out carbon capture and storage, looking after forests and soils, basically from any of these, the challenge is going to be too big. So the proposal that was put forward and is referred to these stabilization or Princeton wedges is that basically we need to mix a number of these renewable technologies and ways of looking after forests and soils, conserving energy and so on. So we piece those together, and different parts of the world will basically come up with different wedges that we do that. But basically within that mix, bioenergy is going to be a very important way to actually deliver a number of renewable sources that we're not going to find very easily or at all from other technologies. So bioenergy can provide a non-interruptible form of electricity, so we're not constrained by the fact the wind isn't always blowing or the sun isn't always shining. It's a good source of renewable heat. We can also produce liquid transport fuels and chemicals to basically replace fossil fuels, and particularly oil in all its forms. And of course we also get the potential benefits in terms of carbon sequestration, in terms of actually carbon that gets locked up or into soils. So there's nothing new, of course, about using land to provide energy. And so if we go back 100, 150 years before the era of cheap oil, we were using land to provide energy, and energy for motive power as well. Of course, the difference now, and going back to John Beddington's talk, is of course we've got the increase in population, we've got increases in people's lifestyle expectations, and of course we've now got also the problem, whether we're working on food or energy crops, in terms of actually dealing with climate change. So the challenge is bigger, but we've done it in the past, and of course we've got all the technologies now that we can use to bring to bear on that problem. So there's a range of strategies we can adopt, including for research in bioenergy. So we can look at identifying new uses for existing crops and residues. And so this is where a lot of the research has been focused initially in terms of generating fuels. Of course, there's some issues with some of that, particularly when we're using food crops, particularly grain crops. There's a big issue there in terms of the food versus fuel debate. An alternative is in terms of developing new crops, and Miscanthus is one of those crops that's been discussed. And of course, within what we do, we need to be spanning this entire bioenergy research pipeline, because we need to ensure that what we're doing in this type of strategic research is actually translatable all the way through. There's no point doing the research if we then find out, well, actually, look, this is going to be flawed at another point along the pipeline. And of course, clearly then we've got to scale up from lab to field to make sure that actually we can demonstrate this at scale. So of course, to do all that, we need a range of integrated research. A lot of this relates to actually plant science research, and then there's other components, which means interactions with engineers, chemical engineers, and also sociologists, economists, and so on, to ensure that we can piece all these things together and actually deliver this. So specifically thinking here in terms of Miscanthus, the challenge that we have here, and in fact the opportunity and the excitement, is actually domestication of a new crop species. So in terms of Miscanthus, for those of you who may not know, it's the Miscanthus that is actually currently being grown, and there's probably something in approximately in the region of about 10,000 hectares in the UK. The crop that's currently grown is Miscanthus giganteus. So this is a naturally occurring triploid hybrid between two parental species. So Miscanthus sacroflorus, sorry, that's slightly blocked out there, and Miscanthus sinensis. So it's a naturally occurring hybrid between a tetraploid sacroflorus and a diploid sinensis. But diploids and tetraploids occur of both those parental species. There's differences in the actual parental species in a number of traits, but the hybrid produces a vigorous hybrid with a number of desirable traits from both the parental types. So Miscanthus is a perennial crop. So it's established at the moment vegetatively because it's a triploid hybrid, so it's sterile. So it's propagated from rhizomes. This grows up over the year and is currently harvested in late winter to spring. And by that time, basically what's being harvested is predominantly carbon. The nitrogen is being remobilized down to below ground. And so the biomass that's taken is largely the above ground stem material, and then that crop regrows. And the crop can certainly, in terms of what we know from trials that have been in place and plots, is that certainly 10, 20 years potentially plus we could have a single plantation without any significant inputs, and particularly of nitrogen within there. So actually, in terms of thinking about what are the targets, what do we want to do in terms of improving Miscanthus? Clearly, and particularly in terms of thinking of this talk, yield is probably the biggest one. So how can we obtain high yields and ensure that those are persistent and stable? There's also discussion about, well, what about the quality of that biomass and how can that be stable and in terms of its composition? The challenge there, of course, is at the moment we don't know what the ultimate technologies will be in terms of utilization of that biomass. So at the moment we know that it's being co-fired predominantly, certainly in the UK, with coal, but in the future there's a range of other technologies, but we don't know for sure what those technologies are. So it's actually difficult at the moment to definitely go down a particular route of breeding for those, although we have an idea of what we might want to do. And of course, whatever we do, we want to ensure that the crop, which is already quite sustainable, that actually is not put at risk. So we ensure that the crop either maintains current or becomes even more sustainable than it is now. But the big one really keeps coming back to yield. We need to produce more biomass on a given area of land, obviously reduce the land take, and therefore the risk in terms of the issues for food security. And of course, clearly targeting land which is not certainly used for primary food production. So in terms of the strategies that we can take in terms of breeding, is clearly at the front end of that we need to collect and characterize genetic diversity. We then can start thinking about, well, how do we use that genetic diversity and potentially use population-based varieties? Or we can create wide hybrids from plant materials that are particularly different, and whether they occur from very geographically separate areas, or try and introgress particularly desirable traits in there. And clearly there's also the biotechnological option as well. So in terms of Miscanthus, it's native to Asia, and so Miscanthus, I guess, is actually very unusual. So it's a C4 crop, as we're hearing from Julian. So that has all the advantages C4 crops have. But actually, as we're hearing in the question, one of the normal issues is actually particularly cold tolerance in C4 crops. So what's particularly unusual with Miscanthus, and why there's quite a lot of interest in it, is it actually does grow all the way from the tropics right the way up into northern China and even into Siberia. So it's actually a C4 species that is able to grow over a huge geographical range. So there's a number of different Miscanthus species, but those that have been of predominant interest is the Miscanthus sinensis, the Miscanthus sacroflorus, which come together to create this Miscanthus giganteus type hybrid, And there's also Miscanthus floridulus, which is a more tropical type, of which there's some interest in as well. And then there's a range of other species. So, of course, there are some Miscanthus species that already existed in the UK and Europe, but basically those had all been largely collected by taxonomists, horticulturalists, for different reasons necessarily than for producing biomass. Of course, the other advantages of going and collecting, and as we're hearing from Sandra and Nat earlier on, is of course if you do that, if you collect yourself, you can ensure that you've got the full record details of where you collected. So of course a number of the materials that already exist, all the information you may have is just China, so you don't really know in terms of what the detailed environment from which that actual accession was collected. And of course the other thing of thinking about biomass is here we're not actually looking at particularly seed production specifically. So actually a lot of the material when you go to collect may not be flowering. So in fact the ability to go and collect vegetative material can also be quite important. So we've been to a number of locations around Asia. So this is in China. So here again, I guess a lot of species in parts of the world you start getting the infringement of agriculture in terms of the impact. So we've been into river systems and collected from small islands where there isn't a lot of agricultural activity to collect those, using boats and so on to get up those river systems, and then collecting material, taking the kind of measurements that we would really want in terms of if we want to try and identify what is their particular value in terms of biomass production. Likewise, we've been into Japan. So Japan, there's a lot of different islands, a lot of these volcanic. So we've been to a lot of those different areas and been identifying different materials, looking for those that may be quite diverse. Taiwan is particularly interested, so it's basically largely subtropical or tropical, and so that we may regard that probably as too southerly for the UK, but what's quite interesting is that you go all the way up from sea level on the island, right there up to almost 4,000 meters in the center. So we've got a huge altitudinal climb, and of course there's a big temperature gradient, and certainly in the tops of the mountains there's snow, and so it's quite a sort of simulated sort of temperate sort of environment. So we've been up into these high altitude areas, and I think the altimeter here is reading over 3,000 meters where we've been collecting material and then actually identifying material. After seeing Sandra's talk this morning, this photograph possibly belongs in the vaults of the Natural History Museum or something rather than in a talk here. But one of the big issues as well, of course, is collecting this material is actually bringing it back and ensuring that you put it through a quarantine process, and particularly the material that's come through as vegetative and before taking it out. But clearly then the issue is, well, taking that out, planting it under, in our case, UK conditions, and looking at a range of traits and saying, right, well, how basically does the plant look in terms of range of biomass characteristics, and potentially how also do those compare when we look back to actually being in when those plants were in Asia. So we're doing some of that work in field. We're also doing some, or getting ready to do some of the sort of high throughput phenotyping using the sort of phenomics facility that's just coming under commission or out of commission in Aberystwyth at the moment, but also using a range of phenotyping methods in the field from actually people going in there to trying to use other methods to automate that process. So some of the work we're doing is under the BBSSC Sustainable Bioenergy Centre, so the Busbeck Biomass Project, where we're looking at a range of ways of trying to understand how basically we may be able to optimize yield in energy crops, so in this case in Miscanthus and also willow. And so this is part here of a combined trial which is both at Ibers at Aberystwyth and also in Rothamsted at Harpenden. So thinking in terms of how practically are we going to increase biomass production and in terms of yield, so one of the ways that we can consider doing that is basically extending the effective growing season. So of course if we've got a canopy that's established longer and it's intercepting more light, the assumption would be that potentially we're going to be able to fix more carbon and we'll end up with more biomass. Of course there are issues with that in the terms of if we increase the time, or in terms of the earliness by which we actually get the emergence of shoots, then of course those shoots, and again this being a C4 crop, they're going to be emerging into cold and potentially frosty conditions. Likewise, at the end of the season, there's going to be the issues in terms of delaying, if we can delay reproductive phase, and particularly if we can delay the onset of senescence, there's potential advantages there in terms of increasing the growing season, but again, we may be subject to lethal temperatures. So this is just an example of actually where some work's been done to compare the date, in effect, of spring emergence with the date of a certain stage of senescence within a collection of 244 different genotypes of Miscanthus in Aberystwyth. And so what this indicates, looking at this here, is that if you have a short canopy duration, so if you emerge late and you flower early or you senesce early, then you're going to be low yielding. But clearly, just because you have a long canopy duration doesn't make you high yielding. And if you look in this box here, you'll see a huge range of yield for actually the same sort of length of canopy duration. So canopy duration on its own is clearly not everything by a long way. So another way by which, of course, we may be able to increase yield is optimize the amount of light that's intercepted and therefore potentially fixing more carbon, accumulating more biomass. And so we can envisage a range of different morphological or architectural forms that the plant can take. And so again, looking amongst this population of 244 genotypes, we've characterized these into a range of upright stems, upright leaves, all the way through to quite sort of sprawling stems and horizontal leaves. And in fact, we can find examples within these 244 genotypes of all these forms except for this last one here. So all these forms here and intermediates of leaf form with the same stem angle exist within that population. So there's potentially a lot of opportunity for modifying or optimizing architectural forms. And this is just an example in the field of actually some of those growing. And again, in terms of the actual leaf area, so actually leaf size, there's a huge variation again within this population. So there's massive opportunities for potentially optimizing those. So another way, of course, is thinking about architecture is also going to be stem number. And again, like pretty much everything we look for, we can find huge variation in stem number. So this graph here basically shows an example where there's two genotypes that have roughly the same biomass yield, but they've achieved it by very different means. So one line here has produced a very large number of thin stems, and another one has produced a very small number, or a small number, of larger stems. So again, that's going to make a difference in terms of how those stems and the leaves attached to them are going to intercept light. So this is an attempt here of actually looking at yield here in terms of dry matter yield and relating that just to, on an individual basis, to a range of traits. So in this case, to maximum canopy height. So the taller the plant relates to, in this case, to well, the maximum canopy height and the tallest stem are obviously quite closely related. But trying to relate any of these to actual yield is actually, again, can be quite difficult. So there's a number of things that we can do by by looking individually, but of course clearly what we want to do is try to stack some of those traits. So this is an example where a number of traits, so in fact 24 traits, have been used to try and actually categorize, in this case, this population of Miscanthus, and we can use those in effect to separate out the different species, so the Miscanthus sinensis, the Miscanthus sacriflorus, so the parental types of the Miscanthus giganteus and those Miscanthus giganteus type hybrids between the two. So we can see that we can actually identify those, but there are exceptions to that, so we find that that doesn't completely explain that. And maybe that's not necessarily too surprising, because in a sense, the way that the Miscanthus taxonomy is actually done traditionally is been in relation to orn length. So that's just taking a single biological characteristic and using that as a way of actually classifying Miscanthus. So this is doing it almost amongst is doing it amongst a much larger number of traits, what all of these really related to in terms of biomass. So clearly where we're going with a lot of this work is how might we match in terms of collections that are made and have been made in Asia to putting them into target geographies, in particular in the UK. And of course, clearly one of the big challenges that we've had over not the last winter, but the 2 winters before that, was very cold winters. So basically, how can we match different areas in Asia to the sort of climatic conditions that we're seeing in the UK? The other thing is in relation to particularly flowering time. So flowering time is actually an important trait in terms of biomass yield, because plants that flower earlier tend to stop vegetative growth and therefore will tend to be low yielding. But like before, late flowering genotypes are not always the highest or high yielders. So we had a project, BBSOC project, which Elaine Jensen was a postdoc on, and she's in fact got a poster outside, I think poster 28. So she's been looking at, over the last 4 years, flowering, including in this population of 244 genotypes. And flowering time across that varies all the way through from June through to November. And in fact, the flowering varies quite differently in the different species as well. So in the Miscanthus sinensis types, that the flowering is occurring from June, and it'll go through to November, and by the end of the year, 80 to 90% of the plants will have flowered. In the case of Miscanthus sacriflorus, only 20 to 30% of the plants will have flowered by the end of the year, and they'll have flowered later. Miscanthus giganteus predominantly will have flowered, so again 80 to 90% will have flowered, but they will have started flowering much later. It's one of the reasons why Miscanthus giganteus produces large amounts of biomass, is it's late flowering, and then goes into senescence. So one of the things we're interested in is, can we relate flowering time to origin, both in terms of latitude and altitude, and those relationships weren't basically very good. However, when we used a geostatistical modeling tool, so kriging, and related that to actual geography, we actually got a pretty strong relationship of the origin of the Miscanthus genotypes and the date of flowering in Aberystwyth. So the plants that flowered earliest in Aberystwyth were occurring here in the northeast of Asia, and those from the southwest of Asia were flowering later in Aberystwyth. So at the moment, we're particularly getting to a point where we're actually mapping some of these, the flowering time in a population which we've just recently created a genetic map for. And so from that, in fact, again, thinking back to stories on we've heard today on polyploidy, the interesting things is that what that genetic map's told us, in fact, is that so Miscanthus is a close relative of sorghum. So sorghum has a base number of 10 chromosomes. Miscanthus is a base number of 19. What's happened is we've had a whole genome duplication, and we've had 2 chromosomes have fused. So we've gone to 19 chromosomes. But of course, what that means now is that we can relate not just the genetics, but also the phenotypes. And so we're beginning to see indications of conservation of some of those, certainly regions of the genome for flowering between Miscanthus and sorghum. So the other obviously issue, and big issue, I guess, in terms of climate change and abiotic stress, in terms of the impact on yield, how are we going to be able to maintain yield in that? So this is taking data from Miscanthus giganteus and modelling it across Europe. So the areas in blue represent frost kill, so there's a 1 in 10 year chance of getting frost kill. In brown, we've got a 1 in 10 year chance of drought kill. The more green, the higher the yield. And so this is looking back, modelling back in time in terms of historical weather, and this is looking forward. In this case, only now 8 years hence, taking the IPCC, not the worst case scenario, the one back from that, so certainly we look like we're at least on that trajectory. And in fact, we can see even in the UK, the southeast is going to become a lot more challenging in terms of Miscanthus, and I'm sure that's going to be the same for whatever food or energy crop we're going to be looking at. So what we've done here is said, right, well, if we can produce a genotype as good as Miscanthus giganteus, but we can bring in some of the traits that we know that we have in the materials we have, particularly in related to drought and frost tolerance, that we can actually significantly change the face of that map, both in terms of now and in the past and going forward into the future. So this sort of climate change proofing of crops we think is going to be really important in terms of driving the breeding we're doing. The other thing that's important, partly in terms of the economics and establishment of the crop, said at the beginning it's established from rhizomes. Clearly, if we want to get new genotypes, new varieties into the market, that's going to have to be, doing that from rhizomes is going to be very slow. Also, the challenge is if we're going to be doing it from seed, how rapidly can we do that? So we've been modelling here in Europe for a range of grasses, including Miscanthus, and looking to see where are the opportunities to do, where can we actually potentially sow Miscanthus, where do we have enough moisture and warmth, and get a plant that's going to be big enough to survive the first winter. So at the moment, that's not really in the UK, but in effect, using a combination of breeding and also agronomic interventions, we believe that we can actually shift that up into the UK. So, of course, the big thing, going back to the beginning again, is how do we scale up? How do we get out of the lab? So we need to set up trial networks, and so we've been doing that with industry. So with Ceres, setting up a range of trial networks. One of the values for that is actually looking at same genotypes or families of genotypes across a range of different environments. So this one here, the bottom set of figures, if you can see, is a range of genotypes that are flowering under conditions in Italy that aren't flowering higher up in Europe. Quality is potentially important, so we need to be thinking about that in terms of the populations we have, and we have variations for that, but we may not necessarily want to be actually making targeted selections for that. Sustainability in terms of the greenhouse gas energy balances are important, and at least understanding that, not necessarily using that to drive the breeding programs, but understanding that. And then really sort of finally, one of the areas that we have a BBSRC case studentship working on looking at some of these bacterial endophytes which occur in Miscanthus, and that certainly have the potential to fix nitrogen. So this occurs in a number of species. Obviously, the rhizobium are the most famous ones, but there's a range of other bacteria that exist, some of which are capable of nitrogen fixation. So we're looking at those at the number of those that exist in Miscanthus. And finally, of course, really in terms of actually to make these interventions, in terms of we need to have, in effect, the genetic variants. So really looking at hereditaries of a number of these characteristics, including dry matter yield, we find that most of these we can explain the variants in terms of through the genetics. There's obviously environmental impact, but clearly breeding is going to be an opportunity to do that. So really just thinking at the end, The big thing, again, is stitching this together. I've really been talking about the sort of plant biology, but we're going to need to stretch that across, interact with other researchers, technologists. We need integrated research. We need integrative teaching. So one thing we're doing is working with the School of Management Business in Aberystwyth. So developed a new MSc in Green Biotechnology and Innovation. And of course, clearly working in terms of public-private partnerships to actually make all these things work across the chain. So that will just leave a set of acknowledgements. There's a number of the people on there that are actually in the room as well. So Elaine's got the poster, and Paul and Kerry, who's quite a bit of the data I've shown, and their names are up, and the slides are also here today. So thank you.
So I'm Giles Aldrich from the John Innes Centre. I was just wondering what the level of uptake currently in the UK is with Miscanthus, and also what you define as marginal land, where you expect it to actually be grown.
Yeah. So, I mean, in terms of, in the UK at the moment, the figure's probably about 10,000 hectares that's currently grown in the field, and that is predominantly being grown and cut and going to be co-fired in power stations. Some of it's going for things like animal bedding, but it's predominantly going for co-firing. So, and then, sorry, the second bit was?
It's marginal land.
Where yeah, no, and that's a fantastic question. I mean, on a whole, what we're largely trying to do is avoid competition with land that's being used for certainly primary food production. In terms of whether we want to actually do it in terms of land classifications, you can do it, but there's a whole debate in terms of what we precisely mean by marginal land. Clearly the issue is not conflicting with those areas of land. And in fact, economics would not drive that, because clearly there's more money to be made from growing wheat than there is growing Miscanthus.
But if you avoid production, if you avoid food crops, doesn't it just put you in competition with biodiversity? Because it's presumably areas that are not currently cultivated that are then marginal.
No, there's areas of land. So I mean, some of the big areas of land that are, or where there's opportunity, even in the UK, that there's where we've had, because of common agricultural policy reform, that actually there's been a huge reduction in livestock numbers. So there's large areas of grassland that are in fact underutilised. So yeah.